Start with Lab 7, which focuses on eukaryotes, particularly fungi, helminths, and protozoa. Mycology, the study of fungi, introduces us to yeasts and molds, all of which are opportunistic pathogens. For example, Saccharomyces cerevisiae is used in bread and wine production, while Candida albicans is the most common fungal pathogen, causing candidiasis, thrush, and irritation. The notable fungi include Rhizopus, Penicillium, and Aspergillus, each linked to specific diseases. Moving on to helminths, we have three main classes: trematodes, cestodes, and nematodes, which are all parasitic worms causing various diseases. Protozoa such as Giardia lamblia and Plasmodium are responsible for significant human illnesses, including giardiasis and malaria. Understanding these organisms and their diseases is crucial for recognizing their impact on human health. Lab 8 introduces the Kirby-Bauer antibiotic susceptibility test, a key technique for determining which antibiotics are effective against specific bacteria. Using a spread plating method, a lawn of bacteria is created on an agar plate. Antibiotic-soaked discs are placed on the surface, and as the antibiotics diffuse, they create clear zones of inhibition where bacteria cannot grow. Measuring the diameter of these zones helps classify bacteria as resistant, intermediate, or susceptible. This method is essential for guiding physicians in prescribing effective treatments. Additionally, various antimicrobial agents like ethanol, Lysol, and bleach were tested, though these are not standardized like the antibiotics. This lab emphasizes the importance of accurate testing in combating bacterial infection. Lab 9 covers epidemiology and the basics of disease tracking. Key terms include morbidity, which refers to the number of people with a disease, and mortality, which is the number of deaths. The Morbidity and Mortality Weekly Report (MMWR) by the CDC is a crucial resource for tracking notifiable diseases in the U.S. Important calculations include incidence rate, which measures new cases, and prevalence rate, which measures total cases in a given time period. The concept of R0 or R0 indicates how infectious a disease is. The lab also introduces the idea of the index case or patient zero, the first identified case in an outbreak. Through a hands-on activity using marcescens and candy, students learned how diseases can spread and how to trace the source. Labs 10, 11, and 12 focus on quantitative microbiology techniques. Serial dilution and pore plating allow for the estimation of viable cell counts, using scientific notation to calculate colony-forming units per milliliter. Mastery of micropipette use is essential for accurate measurements. In Lab 11, students monitored E. coli growth in TSB by taking aliquots every 30 minutes and measuring turbidity with a spectrophotometer at OD 600. This method provides a quick, non-destructive estimate of cell density, as increased turbidity correlates with higher bacterial populations. Lab 12 emphasizes proper graphing techniques, including labeling axes and determining generation time during exponential growth. These skills are fundamental for analyzing and presenting microbiological data.